Hello, my name's Lee. I study Central American pit vipers, and this is a chapter from my PhD where I was looking at the ontogenetic variation in Central American pit vipers. So starting very broad, pit vipers are of the subfamily Crotalinae. They're characterized by their loreal pit that's situated between the eye and the nostril. There's around 250 species worldwide, distributed between Asia and the Americas. And within Central and South America, there's roughly 100 species. So a highly diverse region looking at, and all of which are venomous. So snake venom, the main function of snake venom is prey subjugation, but it can also be used for defense and competition. And again, very broadly speaking, they can be categorized into cytotoxic effects affecting the cells, neurotoxic effects affecting the nervous system, and hemotoxic. And I'm focusing on the hemotoxic side of things, specifically coagulotoxic. So on the right-hand side there, we have the normal human coagulation cascade, where each of these factors will be converted into an activated factor, which will then convert another factor into its activated form, up until fibrinogen is converted into fibrin, and a cross-linked fibrin mesh is formed, trapping red blood cells and platelets to form a strong clot. Venoms have evolved to target this system. They can be either procoagulant, so they're going to activate those factors to form strong clots. They can be anticoagulant to inhibit or destroy these factors, preventing clotting. Or there can be a third one, which is kind of a mix of the two, which is pseudoprocoagulant or thrombin-like, where it's cleaving fibrinogen to form very weak clots that are easily broken down by the body. So it's a procoagulant mechanism, but has a net anticoagulant effect. Within Central America, we spoke about this briefly yesterday, where there's an umbrella term of bothropic envenomation used. So we're lacking sort of species-specific clinical manifestations for all the snakes in Central America. But the known venom effects range from mild to severe local and systemic effects, one of those being coagulopathy. And to treat that, they use the polyval ICP antivenom, which is made up with equal parts Bothrops asper, Crotalus cimus, and Lachesis stenophores. With it being a polyvalent antivenom, they're hoping that the venom phenotypes of these snakes cover a variety of different venom effects. So my study genera, I look at Cerrophidion, or the montane pit vipers, Porthidium, the hog-nosed pit vipers, Metlapilcoatlus, which is formerly Atropoides, they're the jumping pit vipers, and Ophryacus as well, which are the horned pit vipers. This was some work that I did in my honors. So this is a heat map overlaid a phylogenetic tree, just looking at the clotting times of these snakes on plasma. And what was shown was Porthidium were mostly anticoagulant. So warmer colors there indicate faster clotting times, and cooler colors indicate no clotting or slower clotting. Within Porthidium, there was uniquely a procoagulant Porthidium volcanicum and a pseudoprocoagulant Porthidium parasi, but the rest were anticoagulant. Metlapilcoatlus was also pseudoprocoagulant, inducing those weak clots that are characteristic of the pseudoprocoagulant activity. And finally, Cerrophidion had a mix of anticoagulant and pseudoprocoagulant venoms. There's been a lot of work recently done on the venom composition of these snakes. Recently, there was a genus-wide study on Metlapilcoatlus, and there's been a lot of work done on Porthidium and Cerrophidion as well. And what's been found is that the proteome of these venoms are dominated by phospholipase A2s, metalloproteases, and serine proteases as well. There was a little ontogenetic study done as well on Metlapilcoatlus numifer. I think on the left there is the juvenile venom, and on the right is the adult venom. So you can already see that there's a little bit of an ontogenetic shift in venom composition present. And there is also a known ontogenetic shift in the diet of these species. So the main function of snake venom being prey subjugation, a shift in the diet might correlate to a shift in the functional venom activity. Neonates and juveniles are very small, so they feed on lizards, small skinks, orthopterans, and frogs as well. And as they get larger, they can shift to more warm-blooded prey, like small mammals. So these were my study species, the samples that I used. In green there is the juvenile samples. I had adults and juveniles of each of them. Some of them were single specimens, whereas others were pooled samples as well. And I used thromboelastography to look at both the timing and the strength of clots, and the Stego Star Max, which is a hospital-grade machine that uses a magnetic ball bearing system. And then that goes back and forth between the magnets, and then when a clot's formed, the ball bearing stops and the clotting time is automatically measured. So straight into the plasma clotting. With these thromboelastograph tracers, when a clot forms, the trace splits. So that's the SP, that's the split point. And then the clot strength is how far that trace splits. So starting off with Metlapilcoatlus, the adult showed kind of classic pseudo-procoagulant activity where it's fast but weak clots compared to the spontaneous control. And the juvenile counterpart was pretty much overlaid on the spontaneous control. So it wasn't really acting on plasma. With Metlapilcoatlus numifer, the adults showed a lot weaker clots, and they were formed a lot faster than the juveniles. So it looks like it's more of a potency thing where adults are more potent than the juvenile counterparts. It wasn't the focus of this study, but I had both a female and a male Metlapilcoatlus olmec sample that I was testing, and their progeny as well. There were no differences between males and females in plasma clotting. But again, the juvenile Metlapilcoatlus olmec was pretty much overlaid on the spontaneous control and showed no difference there either. In contrast, Ophryacus undulatus, the adult showed an anticoagulant effect. And the juvenile kind of showed a sort of delayed clotting. So we interpreted that as an anticoagulant effect as well. Clots still forming, but quite delayed and quite weak compared to the spontaneous control. This is just another way of looking at the same data, just bar graphs of the clotting time and strength, just highlighting that the juveniles were very similar to the spontaneous control, and highlighting that those clots are quite weak in the pseudoprocoagulant venoms. For the Porthidium species, no discernible clot was formed throughout the entire experiment, so they were all potently anticoagulant in this assay. Moving on to the venoms that clotted plasma, we also tested their ability to clot fibrinogen to determine if they are pseudoprocoagulant. Again, we found that the adults were more potent than the juveniles, and we saw no activity in the Metlapilcoatlus juvenile and the Metlapilcoatlus olmec juvenile. So we determined that there was no coagulotoxic effect in the juveniles of these species. Maybe they're relying on a different venom effect or mechanical subjugation for prey. Onto the antivenom, you can't really see there, but that's, I can't tell what that one is. It's the Cerrophidion Metlapilcoatlus adult. So on the top there is the 8-point dilution curves. And that's illustrated by the area under the curve of the ICP antivenom. And it shows effective neutralization of these pseudoprocoagulant effects on fibrinogen. And then moving on to the percentage increase, that just allows comparison between the adults and juveniles. And it shows that it was effective regardless of life stage. Back to the anticoagulant venoms. This is an assay where we incubate the venom with fibrinogen for half an hour and then add thrombin to it to see if the venom has degraded fibrinogen to prevent clots from being formed. So we showed that all of the Porthidium species were showing fibrinogen destruction, with the juvenile Porthidium eucutaneum being a little bit more potent than the adult. And the same for Ophryacus undulatus. So just looking a bit further into the anticoagulant effects, we looked at the ability of the anticoagulant venoms to inhibit specific clotting factors. And we found multiple clotting factors were inhibited by both adults and juveniles, except for Ophryacus undulatus, where the juveniles showed no factor inhibition across all of the ones tested. So there's quite a clear ontogenetic shift there between the adults, which was inhibiting factor 9A, 10A, 11A, and 7A. I do want to draw attention to the inhibition of factor 7A, where the Porthidium species, both adults and juveniles, potently inhibited factor 7A to prevent clotting, which is a novel activity. I don't think it's been shown in vipers before. Unfortunately, I ran out of venom stocks, factor stocks, and antivenom. So unfortunately, that's where that ended. But it would be really cool to look into that further to test if the antivenom works on sort of a novel venom effect within these snakes. So just to summarize, the ontogenetic shift that we saw was potency-based. They had the same mechanism of action, except for the Metlapilcoatlus, Olmec, and Cerrophidion petalocalensis, where no coagulotoxic activity was observed. Ophryacus showed a cleared ontogenetic shift, where they go from a fibrinogen destructive venom more towards a factor inhibition. The venoms tested were well neutralized by the ICP antivenom, and yeah, the novel factor 7A inhibition across Porthidium was quite a surprise in this study, and it would be interesting to look if that's seen across all species of anticoagulant Porthidium, and if antivenoms neutralize that effect. Thanks very much. I want to thank my supervisors, members of the lab, Sam is in the audience, and the co-authors of the paper.